Hi, everyone. I'm finally getting to you here to the chapter 3 recording. Uh, this chapter is a bit of a challenge because there's just so much to talk about in biological psychology, and there's so many kind of wildly different areas that we could cover um, because we can look at things like what we're going to look at, which is going to focus primarily on the brain and the nervous system and to understand how that works. One of the main reasons I want to focus on that is that there are some aspects of how the cells of the nervous system work, neurons, that can be just a little bit tricky, and that I think the textbook by itself may not really be enough to get that to come across. So I've got my own explanations, and I have found some fairly good, I think, videos from, from of all places, a YouTube channel called 2 Minute Neuroscience. And I think they do a pretty nice job of illustrating some of these processes. And I've got several of those videos incorporated into these slides. But there's other things that we could be covering about that I think are also pretty interesting. For example, in the first slide, I'm just going to give a nod here to the to some issues related to genetics. And so what are the roles that genes play in our behavior? And from there, we could look at things like animal behavior, and a whole lot of biological psychology really looks at animal behaviors so that we can understand what it is that animal behaviors have in common with human behaviors and what can we learn about humans by studying animals. And that's an important area, and it's a very big area with a long history, sometimes a controversial history. And the reason why I'm not necessarily going to go into that in any great detail beyond this, what's on this slide, is because we will actually come back to those topics in other places this semester. For example, there's a whole chapter later on about learning. And a lot of learning research has been done with animals. And so we can focus on that kind of topic, animal psychology, when we get to that chapter, for example. And it comes up in other places throughout the whole rest of the semester as well. So I will focus less on that here, but I do want to focus on this one question because this is pretty important and we may not have much time to cover it in other places. So what you may have heard of before as the nature-nurture controversy is this debate between whether our psychological traits, as I have here, remember back in, I guess that's chapter 2, we were talking about the difference between states and traits, right? And psychologists are interested in both. But when it comes to states, that is these transient things like I'm in a good mood today, or I feel angry right now, or whatever that my state might happen to be, we don't necessarily look at those things and think, oh, that's just your genetic disposition, right? But it's when we talk about traits, on the other hand, things that are more permanent aspects of who you are, or relatively permanent at least, we might think that there's a genetic thing going on. So the nature side of the nature-nurture debate says that it is in your nature to be the person that you are. And by being in your nature, it means you were born that way. That's just who you are. It's your essence, right, that you were born with. And that kind of thinking is much older than our understanding of genetics, right? But the idea of something, of people being born a specific way, is something that's been discussed for centuries. In fact, going back to Greek philosophers like Plato, who argued that we were born with all of the knowledge that we will ever have. So another phrase here is called nativism, right? And nativism is just the idea that that certain aspects of your personality, your intelligence, of your both even physical as well as psychological traits are innate, right? You're born with it. But of course, in modern biology, we would look at that and say, well, it's genetics, right? Your genes have, code for how your body develops. And certainly that affects your physical appearance, your height, your body shape, your facial characteristics that cause you to have some resemblance to your family in an appearance physical way. But it's also important to realize that your genes also determine how everything in your body develops, right? Your organs. And that's why, for example, the human heart and the human liver and the lungs all have the same basic structure and function across all individuals in humans, because we all have the same genetics that specify how those organs will grow and function. And of course, that's why it's useful to study animals when it comes to looking at the cardiovascular system and other mammals, for example, because they have basically the same genetics too that will specify how their cardiovascular systems work or how their metabolic digestive systems work as well. So we can learn a lot from looking at all of that because it's genetic. But let's also not forget about the nervous system, and the brain is growing, and its growth is structured by genetics. And that means that the fact that our brains all look pretty much the same, right? So the structure of the brain, the fact that we have the cerebral cortex and all the same subcortical parts like the cerebellum and the thalamus and the hypothalamus and the brainstem and all of that is the same across humans because of genetic factors that cause the brain to form the way it does. And of course, again, when it comes to looking at animals, a lot of animals have pretty similar nervous systems. Maybe the human brain is quite a bit more complex than other animal brains, but a lot of the subcortical stuff, brainstem stuff, and peripheral nervous system stuff. I'm using a lot of terms here about anatomy of the brain that you may not yet know, but it's later in these notes. But the idea is that a lot of that stuff is the same in other animal species too. Why? Because of genetics. And of course, why is it that that stuff is encoded into our DNA? And that's because of evolution. The idea is that whatever trait that is the phenotype, remember, if you've had biology, you should know the difference between a genotype and a phenotype. The genotype are the genes that you have, but the phenotype are the outward traits that you display. So some phenotype that's coded for by that sequence of DNA has some sort of evolutionary value. It has adaptive value. It has helped our ancestors, whether human ancestors or pre-human ancestors, survive in some way. And so because the genes actually then determine how our brain develops, we have to think about what's going on in the brain, right? Everything that happens in the brain, and it's not just the shape of the brain at a global view, right, of having the wrinkly looking cortex and all that sort of stuff, but it's the actual cells in the brain and how they all have to come together to create the structure that we can see with our eyes. And that means those cells actually have to connect with each other. And there's a junction between the connection of cells called the synapse. And we're going to learn a lot about the synapse in these notes, but the idea is that it is that determines how the brain functions and what it does. And so now we start thinking about, okay, well, what does that mean? That means that what are the phenotypes? What are the traits, the outward traits that we exhibit that reflect the functioning of synapses in the brain? Well, that's our behavior, right? That is our personality traits, our intelligence, our emotions, our capacity for perception, for memory and learning, for reasoning, planning, problem solving, decision making. Every cognitive process that you can think of reflects activity in the brain. And if the brain's structure and functioning is influenced by genetics, then maybe the obvious conclusion is that our cognitive functioning is directly explained by our genetics, right? And our personality and our intelligence is directly explained by genetics. And that's the strong way of stating the nature side of the nature-nurture debate, right? And I've also got this little point here about the focus when you take a nature perspective is the focus on internal causes of behavior. So the idea is that all of the action is happening. I'm going to point to my head in the video here. All of the causes of my behavior are in here, right? What's going on outside of me is more peripheral to the discussion, right? But really everything about me is in here, right? It's not about the world that I live in. It's not about the culture that I grow up in. It's not about my family. It's not about my physical environment. It's about what's in here. That's the nature side, right? But in contrast to that, we of course have the nurture side. And the idea behind the word nurture is that you are not born a specific way, right? So in philosophy, they like to use the phrase, you're born a blank slate, right? That's a bit of an old metaphor that doesn't necessarily quite convey the same meaning that it might have conveyed to the ancient Greek philosophers. But a more modern philosopher had a different version of it, and he was called it the white paper metaphor, right? That was John Locke. And he said something like, this is paraphrasing, that the mind at birth is like a sheet of white paper, right? It's blank, but the pen of experience writes upon it. And so the idea, of course, here is that no, you're not determined, genetically determined to have a specific personality or intelligence or any of that kind of stuff, but your cognitive abilities are defined by your experience. And so when we call it the nurture side of the debate, what it means here is that as we develop from infancy through our early childhood, it is how we are nurtured. It is how we are taught and the world that we grow up in. And it is then a function of our physical environment and our family and our friends and our culture, all of that stuff that shape us. And so here now the focus is on the external things, right? What's going on in here is just receiving the external stuff. We receive and react to the world, right? And we learn how to react to the world. And what we think of as a personality trait, such as a tendency to be extroverted, which is sociable and outgoing and very demonstrative with our emotions, is just a learned response to certain kinds of social stimuli. Or in contrast, introversion, the opposite of that, of being reserved with your emotions, not very demonstrative and preferring to be alone, or at least in smaller social settings, to be retiring rather than outgoing, is again a learned response to environmental stimuli. So because the action now is all in the environment, we now talk about the external causes. Now, historically, this was a debate that was a very fierce debate that focused on nature versus nurture. Some people took that strong genetic perspective and said everything is genetic, and genetic differences explain everything about what it means to be a human or different kinds of humans. But in contrast, other people took the view that everything was environmental, right? Everything is learned. And they literally said you're pure blank slate and there's nothing about you that's predetermined at birth. And it's 100% environment. And that any person can become anything merely by being born into the right circumstance. There's nothing that genetically steers you away from one course of life versus another except your environment. And that's that. But people don't think that way anymore. Scientists, at least, do not think that way anymore, right? So this was a scientific debate in the late 1800s, led by people such as Francis Galton in England, who was falling on the strong nature side of that debate. This led to various kinds of bad ideas in history, because not only was it a scientific debate, but it became a social debate and a political debate. And if you think about what the political implications of something like this might be, you don't have to look very far into our own news cycle to look at people focusing on human differences that might have a lot to do with genetics. And in the late 1800s and the early 20th century even, we had various political movements that focused on a practice called eugenics, right? So eugenics comes in various forms, but it effectively means selective breeding. But what that relates to, of course, is selective breeding means being able to regulate genetic transmission from parent to child. And so if we wanted to eliminate certain undesired characteristics from a population, we could do that through selective breeding to ensure that only certain people were allowed to have children, for example. Of course, there's a stronger way to take care of that, which of course would be something like a genocide, right? Of just getting rid of the people who have the traits that we don't want in a population. But that assumes that those traits are purely genetic. That assumes that those traits would never arise in a person who doesn't have those genes, and therefore would never arise as a function of environmental factors. And nowadays we know that that's not true. All right, so as a scientific level of understanding of this debate, I have here at the bottom of the slide, we consider every trait And I do want to emphasize that every single trait, whether it's a physical trait, such as your height, or whether it's a psychological trait, such as your personality or your intelligence, is the result of a combination of nature and nurture. That's why I have emphasized it there. Every single trait is nature and nurture. There are no exceptions to that. As an example, you might think height is purely genetic, right? But that's not the case, because even though there are genetic factors that will influence, the height of your parents certainly gives you genetics that structures and factors into your actual height as a developing child and how much you grow into your final adult height. But your final adult height requires growth, and growth requires energy. And energy comes from calories in the food that you eat and other, all of the good vitamins and minerals that are required also to support all of the body systems. And if you're not receiving that literal nurture in that case, right, then you will not achieve the maximum height that you otherwise might be able to, right? So again, we see that these environmental inputs will, the genetic tendency is definitely there, right? No one's saying that the genes are not important here, but certainly the nurture input, the environmental input influences exactly how that genetic disposition finally plays out. And that's true for all of our traits, again. And in fact, the modern field is called epigenetics, because what epigenetics studies is the exact nature of gene-environment interactions, right? So we have genes, we have an environment, those two things come together that determine our traits. And there are different ways in which these things will interact with each other. Obviously, sometimes our genetic dispositions can influence the way in which we explore our environment, and therefore that might actually shape the environment we find ourselves in. And that's a gene-environment interaction, because our genes push us into certain kinds of situations. But likewise, there are some aspects of our situations we can't control, and those are going to have an effect on us, which can then alter the way in which our genes are expressed. So that's a two-way street of causality there, right? Genes can affect our environments, but environments can affect our genes in terms of the way genes are expressed. In fact, if we want to get into the rabbit hole of genetics a little bit, we know that genes are not just simple mechanisms that just determine whether a trait is expressed or not. You might learn about in biology class things like dominant and recessive traits, right? Or dominant and recessive alleles that code for different traits. But also some traits are the result of complex interactions between genes, and not just a single allele somewhere that says this is the trait you're gonna have. And these gene interactions can be switched on and switched off. So gene expression can be activated or deactivated. And sometimes there are environmental events that can activate or deactivate certain genetic sequences. It can be an environmental stressor. It can be an illness, a virus. And what we also know from epigenetics is that those kinds of changes to the DNA, it doesn't change the actual genetic sequences, but it changes whether or not they're activated or deactivated. Those can actually be passed on to offspring. And so we see now that sometimes certain environmental events can be encoded into your genetics to the extent that you can pass that on to your offspring. There's research, for example, on how survivors of the Holocaust in World War II experienced such extreme stress that it altered their genetic expression, and that we see these same genetic changes in their living grandchildren. So again, suggesting here that the nature-nurture, the binding there of the nature-nurture interaction, the gene-environment interaction, is very complex, very powerful, and something that actually is then heritable, so that the environmental stuff actually becomes the genetic stuff, right? And so it's got to be everything. It's got to be both nature and nurture, always, right? And that is my take-home message for this particular slide, as we now move on to talking about the neurons in the brain, as promised. So let's talk about neurons, right? These are the cells of the nervous system. And, well, We also have a few other cells, glial cells, but we'll talk a little bit about those. But this is part of the stuff that I was saying at the beginning that is a little difficult to get just from reading the book, I think. So we have approximately 100 billion neurons in the brain and spine. Some estimates are a little bit lower, down around 85 or 86 billion, although I think in some cases that number reflects only just the brain itself. But if we incorporate other stuff like in the spine and the brain stem, we come up to that 100 billion count. But the nervous system is not just neurons. There are also other kinds of cells called glial cells. And in fact, there's lots of different glial cells. There are astrocytes and oligodendrocytes and Schwann cells and a few others I'm probably not remembering off the top of my head here. And they have all kinds of different functions. Some of them, for example, are sometimes described as being the janitors of the nervous system, right? What they do is, so there's something called the blood-brain barrier. And we don't want so of course neurons require nutrients to come from the glucose and the oxygen that's in the bloodstream. But the neurons do not actually directly interface with the bloodstream. That's the blood-brain barrier, right? The reason for that, there are maybe many reasons for that. One, maybe because the immune system would do certain things to the nervous system that we don't want it to. And because the immune cells are floating around in the bloodstream, we want to keep that barrier. And of course, we also have to worry about viruses and stuff that might affect the brain. We don't want to have that. So we keep this barrier. And that's one of the roles of glial cells, right? What they do is they extract the oxygen and glucose primarily from the bloodstream and then transfer it to the neurons. And in doing that, what they also do is they can actually regulate which neurons are receiving the most energy and which ones are not. And in that way, it turns out that glial cells can actually affect the activity in the nervous system, right? So even though the action of our cognitive processes is governed by the neurons, the glial cells can determine which neurons are getting more food, essentially, and more energy, right? So they can influence neural communication. And I have at the bottom of that list here that they may also send in information in ways that are not yet understood. And that's what I mean by that to some extent, is that by regulating which neurons get the most nutrients, they can actually affect communication in the nervous system. And of course, when the neurons are doing their thing, which we'll talk about in a little bit, there are waste materials and byproducts that have to be removed and transferred into our veins, which will then be excreted through the kidneys in the urine. And again, the glial cells play that role. There's also certain glial cells that provide the shape to the brain, right? As I said earlier, the nervous system is genetically structured so that we grow brains that all look basically the same. How does that work? How is it that the neurons know to take on that shape? Well, glial cells kind of provide that framework or structure that helps the neurons kind of attach to this matrix that creates the shape of the brain that we normally think of when we see the brain. And there's also the myelin sheath to talk about here. And we're going to talk about its function here, but there are specialized glial cells that build it. And as you can see in the diagram here at the bottom, it's labeled here. So let's look at the parts here. So this is a typical looking neuron. Actually, there are all kinds of neurons in the nervous system, and they have all kinds of varieties of shapes. But this is the one that's the typical textbook example of a neuron, at least it's one that is relatively understandable. Although I think the basic aspects of the anatomy should always be basically the same. They just may have kind of drastically different shapes in other cases. So let's focus first on the cell body, which is this big part right here, right? That's the main part of the cell, obviously. And in the middle is the nucleus, and the various, the DNA is there, the genetic stuff of the cell is there. And we really don't need to be concerned too much with that. We don't really want to do the microbiology here. But let's look at these weird little branches that are coming off of the cell body. They are labeled here as the dendrites, and as it explains, they are receiving signals from other cells. And we need to get to that point where we understand what those signals actually are. Maybe one way for you to remember what these are is that in, I believe it's Latin, that dendrite means tree branch, and that's what these look like, right? There's also, of course, the cell membrane, which we're going to talk some about, and that's the outer covering of the cell. Now, down at one part of the cell body here is something called the axon hillock, which is this base of the axon, right? The axon being this longer branch that comes off that might look like a dendrite, except it's a bit different, right? In this cell, at least it's longer than the dendrites, which may not always be the case, but in this case it is. But what's also unique about the axon is that it has got this coating on it, right? And it's called the myelin sheath. It's a fatty covering. And as described here, it increases the speed of the signal. So what's happening is that the signals that are received by the dendrites, all of that stuff collects in the cell body, and it collects at the axon hillock. And if that signal is strong enough, it's going to activate a what's called an action potential, which is a new signal that is transmitted down the length of the axon. In order for that to be as fast as it can be, the myelin sheath is going to help speed that up. There are little gaps in the myelin sheath. These are called the nodes of Ranvier. And as described here, there are Schwann cells, which I guess are what's supposed to be characterized by these little dark spots, which are types of glial cells, which produce the myelin sheath. Though you should be careful about that, because Schwann cells produce the myelin sheath for neurons that are in the peripheral nervous system, which means the nerves that move, that go through your body, like the nerves that come off of your spine and go out and down your arms and legs. But in the brain, there are different ones called oligodendrocytes. And maybe you don't need to be overly concerned with all of the little types of glial cells, right? But down at the tip of the axon, we have all these other little branches that, again, kind of might look like dendrites, but these are now called axon terminals because they are the branches and the terminal ends of the axon. And what's not shown here, but it says it forms junctions with other cells, is that this is where another cell and its dendrites would be found. So the axon terminal is connecting to the dendrites of another cell. So then what you should imagine when you look at the dendrites of this cell is that there should be axon terminals from other neurons converging onto these dendrites. And you should now imagine a network of this kind of stuff, right? All of these various little axon terminals connecting to all of these various dendrites, many different neurons perhaps, converging onto this one cell and sending different signals to that cell, which then through its axon terminal might connect to the dendrites of another cell, or might even connect to the dendrites of multiple cells, right? We can think of that as a many-to-many mapping. Many neurons converge to map onto this neuron, and this neuron itself might be signaling many other neurons too. You can kind of get a sense of that in this other picture. All right, here we see the cell bodies with the axons and the dendrites all branching out, connecting with each other. So how do the neuron signals actually work? Well, there's a basic analogy that we can work with, an electrical signal, right? And it has some truth to it, because we're going to be talking about certain ions which have either positive or negative charges. So just like electrons in an electrical signal have an electric charge, have a charge. The ions that are involved in neural signaling also have an electric charge. And so we want to keep that in mind as we go through this. And again, as we have seen here, we're going to receive the incoming, a single neuron is going to receive incoming stimulation in from its dendrites. That signal is going to be carried down the axon, where it then is transmitted to the dendrites of the other neurons. And again, the myelin sheath speeding it up. And one way to think about how the myelin sheath works is that it helps keep the signal contained within the axon in the same way that an electric cord, electric power cord on a lamp or something, has that insulation on the outside to keep the signal contained inside the electric cord rather than shorting it out somewhere, right? So we're going to start by watching first this 2-minute neuroscience video that's going to teach us about
Welcome
basics
to
of
2-Minute
neurons.
Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with the receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
So there we have some of the basics, right? So a couple of things that were a little bit different. Of course, he did say 85 billion for the number of neurons. It's really not, a major crucial fact there. As I said, 85 to 100 billion. But, he also hinted at the idea that there would be dendrites of other neurons found down here at the end of the axon terminals. And he also used another phrase, synaptic buttons. And that's only because the tips of the axon terminals have a little knob-like shape that is reminiscent of a button. And because that's part of the synapse, which we'll be talking about, they can be called synaptic buttons. All right, let's go on here. So we're going to start, let me make sure that that was right. Okay, yeah. We're going to start, now there's a lot of text on this slide, and I've also got a lot of pictures that correspond with it as well. So I got this picture here, I got this other picture here, and I'm going to try to explain a lot of this, but it works a lot better in a live class setting, which is another reason why I've got these 2-minute neuroscience videos, because he can just kind of do that in the video that I won't be able to necessarily do for you. But one of the things that we need to start by understanding here is the nature of the electrical gradient that is inside versus outside the cell. Now, what are we talking about here? A gradient is any unequal distribution of anything in nature, right? Think about something really simple. Think about having a glass of water and you have a dropper full of food coloring, some blue food coloring, right? And you put that little drop into the glass of water. And the first thing that you'll see is that all of that food coloring is concentrated in a single little droplet at the top of the glass. That's a gradient, right? Because it means that that food coloring is unequally distributed inside the glass of water. But one of the things that you'll find is that it quickly starts to distribute and spread out all inside the glass. And after some period of time, it spreads all across the glass. And eventually now you should have, of course, if you stir it up, you help it along. But the idea is that ultimately the dye becomes equally distributed across the glass. And that's something that relates to the idea that this is something that just happens naturally in nature, that nature doesn't like to have gradients. It likes to even things out. that's actually related to thermodynamics, right? The entropy, right? Second law of thermodynamics. We increase entropy. You might have also heard the phrase nature abhors a vacuum, right? What's a vacuum? A vacuum is a gradient, because if you have a situation where you've got one area of space that has no air and another area of space that has air, that's a gradient. That's an unequal distribution of air molecules. And so what's going to happen is, if you can only maintain that if you keep up some sort of barrier, right? But as soon as you remove the barrier, all the air that's in the place that has the air is going to rush over to the place that has less air, right? And that vacuum will be destroyed, right? That's how weather works, right? High pressure and low pressure systems represent gradients of pressure. And the reason we have wind is because all of the air is rushing from high pressure areas to low pressure areas, right? And that's wind. And the same thing happens here with neurons, is that now we have a gradient, but the gradient has to do with ions that have either a positive or a negative charge. And so what's happening here is that there are various ions inside the cell, as it's described here. There are some protein ions. They have a negative charge, and there are some potassium ions that have a positive charge. So you got some things that are both positive and both negative. But by and large, the total proportion of ions inside the cell tends to weigh more heavily towards these negatively charged ions, all right? But outside the cell, we have other stuff. We have some chloride ions, which have a negative charge, and we have some sodium ions that have a positive charge. And on the whole, the proportion and distribution of ions outside the cell tends more towards the ones that have a positive charge. So now we have an imbalance or a gradient. There's a little bit more negative ions inside the neuron and positive neurons outside the neuron, so there's an imbalance. And that creates this state called polarization, right? Where this unequal distribution of positive and negative charges that is keeping more of one kind of neuron or one kind of ion inside versus outside. And just like we talked about with the vacuum example, if you keep a barrier between the vacuum and the rest of the air, you're going to maintain the vacuum. And so we have a barrier here. It's the cell wall, right? The cell wall can stay closed and not let any ions move back and forth between the inside and the outside of the cell. And that maintains this polarized state. It maintains the gradient of negative and positive ions. But of course, what might happen, just like when we remove the barrier for the vacuum state, if we remove the barrier here, which means to open up some pathways in the cell wall, the gradient is going to try to undo itself, right? Positive and negative ions are going to start moving back and forth to equal out the charge and the distribution of these ions. We've got another 2-minute neuroscience video here. I'm going to come back to that slide. But this actually illustrates the issue of the electrical gradient.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss membrane potential. Membrane potential refers to the difference in electrical charge between the inside and the outside of a neuron. This is the plasma or cell membrane of the neuron. It separates the inside of the cell from the outside environment. We'll say this is the inside, and this is the outside of the neuron. The difference in electrical charge develops due to the grouping of ions on the inside and the outside of the membrane. Ions are atoms that have either lost or gained electrons, and thus have a positive or negative charge. There are several ions that play an important role in the membrane potential of neurons. There are positively charged sodium ions, represented by these blue circles, and negatively charged chloride ions, represented by these green circles. When a neuron is at rest, the sodium ions and chloride ions are more prevalent outside of the cell. There are also positively charged potassium ions, represented by these yellow circles, and various negatively charged ions, often referred to as organic anions, represented by these gray circles. Anions.
Just pausing it real quick here. So, of course, maybe you should be reviewing your chemistry here to make sure you remember the periodic table and the chemical symbols: Na for sodium, K for potassium, Cl for chloride, and also what ions are in terms of charged atoms, in terms of having lost an electron or gained an electron, and why they would then have a positive or negative charge. But we're not going to worry about that. but he mentioned it briefly there.
Is simply a term for a negatively charged ion. When a neuron is at rest, the
Oh, and by the way, those organic anions that he's talking about here that are inside the neuron, those are the same as the protein, negatively charged protein ions that I had mentioned on the previous slide.
potassium ions and organic anions are more prevalent inside the cell. At rest, the inside of the neuron is more negatively charged than the outside, causing the resting membrane potential of an average neuron to be around negative 70 millivolts. One way this potential is maintained is through a mechanism known as the sodium-potassium pump. This is a transport protein that uses energy to constantly pump 3 sodium ions out of the cell, while at the same time pumping 2 potassium ions into the cell. Because there are more positive ions being pumped out than in, it helps to keep the membrane potential negative. Unlike other ions, potassium tends to move fairly easily across the cell membrane through ion channels, which are membrane-spanning proteins that allow ions to pass through. Potassium will pass out of the neuron until it reaches the point where it is at an equilibrium, when forces like diffusion aren't pushing it in one direction or the other. At this point, the membrane potential of the neuron is around negative 70 millivolts, which is known as the resting membrane potential.
I'm going to go back just a little bit here.
Negative 70 millivolts, which is known as the resting.
Let's have a look at this picture real quick here. So he's adding a little extra detail in there that I did not say. So as I mentioned, we have this with this unequal distribution of charged ions inside and outside the neuron. And as I said, we'll keep a barrier in place to keep it that way, right? But in reality, the cell wall of a neuron is not a perfect barrier. And there are these things called ion channels, which is this blue little hole like in the cell membrane. And potassium has a tendency to leak out, right? And it's going to want to leak out because the potassium has a positive charge, and because there's too much negative electricity inside the neuron, that those positively charged ions want to move away from it to go outside and to equal out the distribution. So when he talks about a resting potential of negative 70 millivolts, what that means is that the sum total of all of the electrical charges of the ions inside the cell is about 70 millivolts less than what's going on outside the cell. And that is the gradient, that's the polarization. And if we wanted to equal that out, just like we talked about with the air moving between a vacuum and a non-vacuum space, you would end up with a 0 millivolt difference between the inside and the outside of the cell, right? So that negative 70 means that we're keeping more negative ions on the inside or fewer positive ions on the outside. So as these positive potassium things are leaking out to try to restore balance here, that's going to move us away from that negative 70 millivolts. And the neuron is actively working to counteract that. And that's what the sodium potassium pump is doing. The sodium potassium pump is going to keep pulling that potassium back in. Even though it leaks out, it's going to pull it back in. And meanwhile, what it's also going to do is it's going to try to kick out some sodium ions too, right? And so it tries to keep the sodium on the outside of the cell, and it tries to keep the potassium on the inside of the cell. And because the potassium is usually always leaking out of the cell, it's continuously working to bring the potassium back in. And so he did say that this requires energy, right? So that's one way in which neurons use energy, is because the sodium potassium pump has to continuously work to keep this state of affairs at this resting potential of negative 70 millivolts. Now back here. Now what happens when, now that resting potential means that the cell is at rest. So even though the sodium potassium pump is doing some work, the bulk of the cell itself is not necessarily doing anything. It's not going to do anything until it gets activated. And how does the neuron get activated? The neuron gets activated because a signal comes in from the dendrites, right? That's the input coming in from those other neurons. So what happens? We talked about it being an electrical signal, right? But what happens when, and that's why we're talking about ions, positive and negatively charged ions. So what happens when the cell comes in from the dendrites? Well, this is going to cause these gateways in the cell to open up, not just like the one that we just saw, but there are other specialized gateways that are called sodium channels. And these are special channels or gateways or doors in the cell membrane that only let sodium ions pass through, because sodium ions actually have a particular shape and they can only fit, only sodium can fit through these things. So what's going to happen is that the sodium ions have a positive charge. And because the sodium has a positive charge and because the voltage inside the cell is negative, the sodium is going to want to flow into that negatively charged space to raise its voltage up, right? Because now we want to do that. We want to eliminate the imbalance between the negatively charged inside and the positively charged outside. And one way to do that would be to let the sodium come into the cell. But that can only happen when these sodium channels open up. And the sodium channels only open up if the signal from the dendrite opens them up. And that's what the incoming signal actually does, is it opens the sodium channels and lets the sodium come rushing into the cells, right? So let's have a look before we finish that slide. Here is another picture that shows things such as the typical distribution of potassium, mostly inside, a little bit of sodium, but sodium mostly outside and a little bit of potassium. And there could be a sodium-potassium pump that's continuously trying to keep it that way. But what's happening here is that when a signal is activated, these sodium channels, which are the ones highlighted here in magenta, will open up. And that's going to let the sodium start coming into the cell, right? And that's what's happening here in this graph. We see that there is the resting state of -70, which is what this blue line started at. But when the signal is happening, there is this period where the sodium channels begin to open. That's at 0.1, down here at the bottom, it's coded here. That tends to be happening at around -40. So the voltage is gradually increasing. And then suddenly those sodium channels start opening up at a drastic rate. And this causes a massive influx of sodium ions into the cell. And that causes the voltage of the cell to rapidly increase. At which point, at this peak, something new happens. The sodium channels close. And now the potassium channels open up. So now that we have a positive charge inside the neuron at point number 2, because now there's a whole bunch of sodium inside the cell, if we open up these potassium channels, the potassium is going to start leaving the cell because it's positively charged and the outside of the cell is negatively charged. And so the potassium is going to want to go towards that negative charge. And that's what's going to happen here, is that now at that point, at point 2 in this graph, as all of the positively charged potassium ions start leaving the cell, the voltage is going to start decreasing again. And again, it shoots way down, way below the original resting state of -70, right? It goes all the way down to -90, as it says here. And then the potassium channels are going to close. And now what we're left with is a state where we're kind of at the opposite of where we began, because now the cell is full of sodium on the inside and potassium on the outside, and we want to restore it back to its original state. And that's, again, where the sodium-potassium pump in point 4 here, sodium-potassium pump here, is going to have to get working again to start pulling the potassium back in and kicking the sodium out. And it takes a little bit of time for that to happen. And this is called the refractory period, where it takes a little bit of time for the neuron to move from this -90 millivolts back to its resting state of -70. There is another picture here that illustrates the same principle, that shows that we're at -70 at rest. But if a signal comes in from the dendrites, it's going to open up some sodium channels. And if it's a weak signal, this voltage is going to come up. But if it's weak, it might not be enough, and it might come right back down to its resting state. But if enough of those sodium channels open up that cause the internal voltage of the cell to cross this blue line, that's called the threshold, of the firing threshold, right? And as described on the previous slide, that would be typically around -40 millivolts. And so if it's enough to cross that threshold, then that causes this cascading effect of multiple sodium channels opening. And again, rapid depolarization as we move from -70, -40, up past 0, all the way up here, where the sodium channels closing and the potassium channels are opening. And now the potassium leaves the cell as we drop way back down below the original resting potential. Everything closes, and then we have to use the sodium-potassium pump to restore back to our original state. So if I come back here to our previous slide, that's what was showing here. Sodium channels are opening, and we get this rapid depolarization as the cell's interior becomes positive. So then what's going to happen is we're going to close the sodium channels, potassium channels open, everything switches sides. Sodium is now inside the neuron, potassium is the outside. And then gradually the sodium potassium pump has to switch everything back to the original state, and it's ready to repeat. So that voltage spike, and that's what we're looking at here in this graph here, is a change in the voltage of the neuron from negative 70 millivolts all the way up to nearly positive 40, and then back down to about negative 90 before returning to its resting state. That's a spike of the voltage, and that is what's called the action potential. And it's this voltage spike that starts at the axon hillock. If enough sodium all collects into the cell body and at the axon hillock, that then triggers the signal to travel down the length of the axon, where it will then stimulate another neuron. So I've got a video for this. Whoops, I went right past it. Let's have a look at this one.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the action potential. The action potential is a momentary reversal of membrane potential that is the basis for electrical signaling within neurons. If you're unfamiliar with membrane potential, you may want to watch my video on membrane potential before watching this video. The resting membrane potential of a neuron is around negative 70 millivolts. When neurotransmitters bind to receptors on the dendrites of a neuron, they can have an effect on the neuron known as depolarization. This means that they make the membrane potential less polarized or cause it to move closer to zero. This chart shows membrane potential on the y-axis and time on the x-axis. When neurotransmitters interacting with receptors causes repeated depolarization of the neuron, eventually the neuron reaches what is known as its threshold membrane potential. In a neuron with a membrane potential of negative 70 millivolts, this is generally around negative 55 millivolts.
So he's using a slightly different number there. I had previously said negative 40. He said generally around negative 55. What's important to note is that the actual threshold for a neuron is not always the same number. So in this example, he's going to use negative 55. So don't get caught up in that little detail.
When threshold is reached, a large number of sodium channels open, allowing positively charged sodium ions into the cell. This causes massive depolarization of the neuron as the membrane potential reaches zero and then becomes positive. This is known as the rising phase of the action potential. The influx of positive ions creates the electrical signal known as the action potential, which then travels down the neuron. Eventually, the action potential reaches its peak. Sodium channels close and potassium channels open, which allows potassium to flow out of the cell. This loss of positive potassium ions promotes repolarization, which is known as the falling phase of the action potential. The neuron returns to resting membrane potential, but actually overshoots it, and the cell becomes hyperpolarized. During this phase, known as the refractory period, it is very difficult to cause the neuron to fire again. Eventually, the potassium channels close and the membrane returns to resting membrane potential, ready to be activated again. The signal generated by the action potential travels down the neuron and can cause release of neurotransmitter at the axon terminals to pass the signal to the next neuron.
Well, and there we have it. So again, the idea of the signal causing the depolarization as the sodium rushes in all the way up to our positive peak, and then the potassium rushes out. We end up way down below in this hyperpolarized state, and then the sodium potassium pump works to bring everything back to normal, to the resting state, and it's ready to repeat at that point. Neurons can actually repeat this process of firing an action potential about 1,000 times a second at their fastest, right? But they don't always fire 1,000 times a second. Only when they're receiving continuous excitation do they fire 1,000 times a second. Sometimes they might only receive a medium signal. They might fire 500 times a second or 100 times a second. Or sometimes they are actually, and this is something else to talk about, sometimes action inputs from the dendrites, instead of being excitatory, are actually inhibitory. And what that does is it pushes the internal voltage of the neuron down below its resting state and actually is actively working to prevent the neuron from firing at all. And so it fires 0 times a second as a result of that. So one thing that we want to also understand here is that the action potential is actually a traveling signal, right? It travels down the axon, right? As the sodium and potassium channels are activated, not all at once in the whole neuron, but in a sequence. down the length of the axon. And this is one reason why the myelin sheath actually makes it faster. So imagine, for example, if you have an entire axon, let's imagine a gigantic neuron, and you have an axon that's 100 feet long. That's not real, obviously, but just imagine, right? Then what would have to happen is that down the length of this entire 100-foot long axon, sodium channels would open at the beginning, sodium would come in, and then 1 foot down, the potassium channels would open up and the potassium would go out. And then 1 foot down, the sodium channels would open and sodium would come in, and then the potassium would go out. And you'd have to keep going 1 foot at a time, all 100 feet down. And it would take a lot of time to go 100 steps down this 100-foot long axon with sodium and potassium going out. And so imagine what would happen if you put the myelin sheath around it. What happens there is that the signal is allowed to jump down the axon wherever there's the myelin sheath. And then when you hit those little gaps in the myelin sheath that we previously saw called the nodes of Ranvier, that's where the signal gets refreshed with new sodium and potassium channels before it then jumps down another sequence of myelin. In classroom settings, sometimes what professors like to do to illustrate this is to have people stand side by side. So imagine now, instead of a 100-foot long axon, imagine 100 people standing side by side, 1 foot apart. So that now we have 100 feet of people. And we give them a baseball. And the goal is just to pass the baseball down this chain of 100 people. So one person has it and they have to pass it to the next, who then passes it to the next, who then passes it to the next. And we have to go 100 steps of passing the ball from person 1 to person 100. And you can imagine that's going to take a little bit of time. Right? It's not going to take, it's going to take, you know, at least 100 seconds, right? Assuming it takes only 1 second for one person to receive the ball and move it to the next. Might even take a little bit longer, right? But now imagine we can take some of the people out of this lineup and we can space people 10 feet apart. So now we only need 10 people. And so one person has the ball and that person throws it to the next person in line, who catches it and turns and throws it to the next person in line, right? So now you don't have to have step, 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 step 100 times. You can skip steps. You can just jump from one person to another, throwing the signal from one gap in the myelin sheath to the next. And that's how the myelin sheath actually speeds up the signal, right? As it lets it skip down steps. We're removing some of those pathways in the axon and replacing it with the myelin sheath so that it can just jump down from one node to the next. But at the end of the axon, something different happens, right? So that signal that travels from the axon terminal to the dendrite of the next neuron does not involve the sodium and the potassium. It involves a different chemical, neurotransmitters as labeled here. So these neurotransmitters are special chemicals that will be released into the synapse being the junction between the axon of one neuron and the dendrite of the next. So neurotransmitters as described here are chemicals that are contained in little packets called synaptic vesicles. You might think of them as little bubbles, right? Little bubbles that contain molecules of neurotransmitters. And what's going to happen is that as the action potential travels down the axon, when it reaches the tip of the axon, it's going to cause these synaptic vesicles, these bubbles of neurotransmitter, to bind to the cell membrane and dump the molecule into the synapse. Specifically as labeled here, the synaptic cleft, the gap, the actual gap between the neurons. And it is these neurotransmitters and what they do to the dendrite that is next thing that we have to consider here. So what's happening here is that the dendrite, on the outside of the dendrite are little receptor sites. And neurotransmitters will activate very specific receptors on the dendrites that causes that neuron to either become excited and then open up those sodium channels and fire a new action potential. Or as I talk about, sometimes neurotransmitters can activate receptors that are actually inhibitory, that might actually push more or pull some of the positive ions out, really, that even pushes the voltage of the neuron further below its resting state, preventing it from firing. And actually, sometimes neurotransmitters can cause metabolic changes. So it can activate certain receptors on the dendrites that cause growth processes on the dendrite, right? So that could cause something like to build more dendrites, right? To branch them out, right? So that's one thing that you want to remember here, is that our brains are not set in stone. We have a principle called neuroplasticity, that our brains change over time. Our brains change as a function of learning. Hence, think back to the nature-nurture issue here. Environmental inputs, learning actually can change the brain. So even though the genetics cause the brain to develop a certain way, the environment changes the brain as well. Learning changes the brain. Learning can cause the dendrites to branch off and create more extra branches, to grow more receptor sites on the tips of those dendrites, can cause axons to grow longer and to branch off a little bit more and connect up to other neurons, right? And think of these receptors and the neurotransmitters as something that works like a key in a lock, right? So only certain neurotransmitters can activate certain receptor sites, because the various neurotransmitters that we have in the nervous system are all kinds of different molecules, and they have different molecular shapes. And the receptor sites on the dendrites are like the locks that will only accept a certain molecular shape to activate them. And so you have to have just the right neurotransmitter to activate just the right receptor on a dendrite. And that's going to cause a very specific response in what's called the postsynaptic cell, that is the receiving cell, right? Whether that's an excitatory response or an inhibitory response, or maybe even a growth metabolic response. After this process is complete, we have to clean up the synapse. You don't want to leave the neurotransmitter in the synapse where it might continuously have some effect on that postsynaptic neuron. You don't want to continuously excite a neuron. That actually causes something called excitatory neurotoxicity. And when people have seizures or when people have a stroke, sometimes you end up with something called neuronal excitotoxicity, because the sodium channels are continuously open on neurons and they're repeatedly firing. And then those neurons are flooded with so much synapse that they actually die. And then you end up with a whole bunch of dead neurons. And then, of course, you end up with the brain damage that results from sometimes seizures or strokes and whatnot. So we have to clean up the neurotransmitters. And this is a process that might involve what's called reuptake, where the axon itself sucks the neurotransmitter back up. So that's called the presynaptic cell. Reuptake by the presynaptic cell is basically the axon sucks the neurotransmitter back in. But sometimes the neurotransmitter breaks down and has to be cleaned up. And this is where, again, where the glial cells doing their janitorial duties work to get rid of all of these byproducts of the neurotransmission and then excrete it ultimately through the kidneys. Here's a picture that helps us visualize all that. So up top, we again see the presynaptic cell with its axon sending the signal following the red arrow down to the dendrites of the postsynaptic cell, which is then receiving that. And this is the magnified view. We can see the axon terminal, the terminal button, as we had previously called it, right? And these little bubbles floating around inside the terminal button, full of these little green molecules of neurotransmitter. And when the signal arrives at the tip of the axon terminal, you can see that these bubbles now bind themselves to the cell membrane, where they can open up and dump the neurotransmitter into the synaptic cleft, this gap, where they now activate these very specific receptors on the membrane of the dendrite. And we can see here, again, it kind of like works as a key in a lock. And if this neurotransmitter is the right key to open the lock, then we can see that this is going to let this is maybe confusing because it's showing potassium, but let's just focus on the fact that we're going to let the sodium in, right? It's a sodium channel. We're going to let sodium come rushing into the cell, right? And then eventually the gate or receptor lets go of that neurotransmitter, right? And kicks it off. And that's when we have to clean it up. We have to either reabsorb it back into the axon, or glial cells will take care of it. And we have another 2-minute neuroscience video for
Welcome
that.
to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
And so what the video is leaving out there in its brief 2-minute duration is that diffusion leads to those molecules being collected by glial cells so that they can be removed. And the enzymes that break down the neurotransmitter also are coming from glial cells. Well, this is where we're going to stop this video as we move from talking about the small stuff in the nervous system, the neurons, the cells, to talking about the bigger stuff. So when we talk about gross anatomy, we're talking about the parts that are identifiable that we can see with our eyes, right? So let's stop it here, and we'll break this video into 2 parts.